Okay. Should I start right again, or did people hear the beginning? But anyway, there's my title. We'll move along to not lose um, more time. And thank you for your patience. Sorry about that. Um, so yes, looking at major evolutionary innovations, and one of these would be viviparity and the origins of live birth, which is something that has occurred across many, many different taxonomic lineages in a repeated evolutionary way. And a thing about major evolutionary innovations or major evolutionary transitions is that they tend to lack these transitional states. They're kind of a step change in evolution. And the origin of viviparity from ancestral oviparity is an example of this. There's an absence of intermediate forms, usually, so we have this kind of great leap of evolution. And that requires concerted, coordinated genetic changes and evolutionary changes happening along with the complex phenotype. So it's a package that has to go through a sort of fitness valley. And viviparity is a very extreme example of this. And so often these major evolutionary innovations are old and a bit difficult to reconstruct. And that has hampered our ability to infer the genetics of these traits. Today I'll speak about an example that we've been studying that is in squamates that has been able to look at this in more recent evolutionary terms. So the two major alternative reproductive modes that we have in amniotes are, of course, we have a viviparity, so also called live bearing or live birth. So this is when the offspring comes from the mother fully formed as a neonate, but as an independent living individual. Well, oviparous, of course, is egg laying. And so there's a period of external incubation within an egg. And this phylogenetic tree just shows that, of course, when we think of live birth, we always think of mammals and ourselves. But in fact, there has been the origin most abundantly to viviparity has happened in squamates. And so this tree is just to show that from this ancestral oviparous ancestry of all amniotes, we have just one origin, in fact, of viviparity at the base of therian mammals. And in squamates, we have perhaps, people argue many times about exact numbers, but could be something more than 100 independent origins of viviparity. And so within the squamates, which are the lizards and snakes of reptiles, so we have many, many different forms, the squamates being famous for this loss of limbs and very elongate forms, but then organisms that inhabit a great variety of different environments, going from extreme dry to aquatic forms. And so there's a great diversity within squamates, approximately 10,000 different species. And these origins, independent origins of viviparity, have happened many, many times across the tree. And so this was this Perrin and Burbank paper from some years ago that sparked a lot of controversy about the origins of viviparity and oviparity. So I'm not using it from that perspective, but I am there. They did have a very detailed reconstruction of these independent origins. And so here the oviparous and viviparous, you can see with the different blue and red colors, have been many, many different origins across this really rich tree. And we know that viviparity also in amniotes, so in fishes in particular, but also in amniotes, is associated with elevated diversification rates. And because it's releasing some of the environmental pressures that can maybe constrain this diversification. So with viviparity, lineages diversify into many, many different new niches, and they have statistically elevated diversification and speciation rates. And then it also is associated with different life histories. So there's extensive theory about the effect of viviparity on reproductive rates, for example, and maternal investment, which I'm not going to go into today. But then there are also biogeographic associations. And so we see across the globe a clear association between viviparous squamates and cold climates. So well, in the tropics, there can be both oviparous and viviparous lineages. Once we get into the temperate regions, like we have here ourselves in Glasgow, even we have viviparous squamates here. And so it's associated with, it's one of these major transitions that is then associated with many, many different ecological and evolutionary aspects. And I have this card which I found from children's school about Montessori. So when we think on the one hand of being oviparous or being viviparous animals, I mean, this seems like child's play. You can either have eggs or have live birth, and it's, it doesn't seem like that big of a difference. Either the baby is in the mother or all the way to birth, or in the baby, when we think of it in those terms, is inside of an egg. But in fact, there's a huge suite of physiological and behavioral modifications that happens with the transition between one reproductive mode and the other. And this is almost always from oviparity to viviparity. So, one of the key aspects is this how nutrients, water, and gas exchange happen from the mother to the offspring. So in viviparity, there's connections in the more complicated forms of viviparity. There's connections between the mothers and the embryos that facilitate this exchange of gas and water and nutrients as well. And this extensive the extent of this complexity depends on different types of viviparity. So, viviparity spans from quite simple viviparity, as we'll talk about today, so being lecithotrophic, so related almost to an egg within the mother, all the way to full, what we think of as the most complex form, because it involves the most new organs, is placental viviparity. And then there's, of course, egg calcification or not. And so this is believed to be part of the issue with this being a major transition between oviparity and viviparity, is that the calcification of the egg allows the egg external to the mother to have free gas exchange. And being intermediate in these traits is really shown to be a fitness valley. But this matters not just for thinking about the covering of the egg, but actually the bones of the developing embryo draw calcium from that eggshell. And so if we don't have an eggshell, it has to get calcium in a different way. So it requires a really rerouting of a lot of complicated physiological events in the mother. And these have to be in a very tight time frame. Then, of course, the duration of pregnancy, the conditions of incubation, developmental rate of the embryo may differ. And then the immune response is extremely complex and relates to the types of viviparity and also the viviparity and oviparity. So to say there is a huge suite of phenotypes that are related to this one sort of mode step change. And despite the fact that all amniotes are either oviparous or viviparous, so far the genetic basis of this has been really unknown. And the problem has been is that there's really deep genetic divergence in most cases between the the diverged lineages. So it's not possible to do the kind of experiments that would let these traits be dissected between the genotype and the phenotype, so to speak. So today I'm going to talk about a study that we were doing looking at the genetic basis of these alternative reproductive modes. So the same as parity modes. Sometimes I say parity, sometimes reproductive. Oviparity and viviparity. And this was quite a group effort. Really the heavy lifting led by Hans Recknagel, who did his PhD on this work and also a postdoc on this project. And then a suite of other collaborators from within Glasgow, and Maureen Bain is a specialist in eggshell development. And Andrei Yurchenko and Madeline Carruthers contributed to other aspects of the genomics. Mosen contributed to the embryology, and Nick Caminos to the biogeographic patterns. But I'm happy to be able to present it today because many years we've been working on this particular study. So today I'm going to talk about demonstrating some phenotypic differences between these alternative reproductive modes. So in this case study lizard, and identifying and characterizing these interparity hybrids, which was pretty exciting. And then infer the genetic architecture of reproductive mode based on these key traits. And then looking for some examples from gene expression and looking at signals of selection on this reproductive mode divergence. And then also looking at shared genes of pregnancy across vertebrates. I'll touch on these last 2 points at the end. And then one of the key aspects here is that in this experiment I'm going to talk about today, we really have the chance to look at these things while holding the environment constant, because we're looking at a case of oviparity and viviparity without the noise of environmental differences. So usually looking at comparing even closely related oviparous and viviparous taxa, they're usually found in different locations. And so there's a lot of environmental and evolutionary variation to be considered. So here we can remove those and really just look at the genetic and phenotypic relationships. So our model organism for this is called the common lizard, but I don't think it's so common. I think it's pretty amazing because it's got actually 2 different reproductive modes bimodally within the same species. This is Zootoca vivipara. And so it's a lizard that you would find outside of Vienna, and you'll also find outside of Glasgow. And some lineages are egg laying, and so the mother lays a clutch of eggs with embryos within them, and they develop externally for the last stages of incubation, and then of course hatch. And then the other lineages are viviparous, and so these little neonates are what is sort of birthed from the mother as a clutch without any of this external incubation phase. And we had recently completed an analysis, and also other researchers have shown that this is probably the youngest transition to viviparity in squamates. So we're looking at only about 3 million years divergence. Whether that's young or old depends on one's perspective, but amongst all squamates, this would be the youngest transition that is known that has fixed differences between species. And quite amazingly, some years ago, it was proposed that there was this hybrid zone. So in the Alps, there's contact regions between the oviparous and the viviparous, and usually their distributions are non-overlapping. But there was found to be an overlapping site, which was published from Werner Meyer at the Natural History Museum in Vienna. And it was suggested that there might be hybrids, and this was based upon observing a couple of phenotypes in females that seemed not quite one or the other, but had really not been even scratched the surface of it. And so this work was initiated with Werner Meyer, who has since passed away, but he was instrumental in the early parts of our project. And people had looked for hybrids in other locations throughout the Alps and not ever found any, looking for either genetic or phenotypic analysis. And so, yes, as I said, this could be a lizard that you would find here in Glasgow, or you could find it in Vienna, or else distributed through all of Eurasia. It has the broadest distribution of any terrestrial reptile, and it goes all the way up to the Arctic Circle, and it can be found at high elevations and low elevations. So it is really a very resilient lineage that has been very successful. And this species is made up, as I said, of these different lineages. Some of them are viviparous and some of them are oviparous. And so it's ancestrally oviparous, and as its the lacertids, the group of lizards it's from, is also ancestrally oviparous. And so we have an ancestral oviparous lineage, and then we have these derived viviparous lineages. And there had been quite extensive discussion over the last 20 years about exactly the timing of these events and the distinctiveness and whether there was multiple origins of viviparity. So we published a phylogeny on this a few years ago, which showed that the best supported phylogenetic analysis was that there was in fact a reversal to oviparity in one lineage, and an ancestral oviparity, as I've said, and then a single origin of viviparity, and was most likely followed by a reversal to oviparity. And so actually this is another project that we're working on right now, looking at differences between these two oviparous lineages. And today I'm going to speak about the study focusing on the eastern viviparous and the central viviparous too. Okay, so we were looking at in detail at this contact zone, as I said, between the oviparous and the viviparous, which is in the Gaul region of Austria, in the Gautal. And it's the viviparous lineage is coming up over the mountain and moving down the side, the other side. And from the valley below, sorry, did I say viviparous? That's what I mean. And from the valley below, the oviparous is coming up. And there's an extremely narrow region. So we're talking about really at one sort of field where they come into contact, and there's these putative hybrids. And so over several years of intensive sampling, we've been sort of trying to elucidate this contact and hybrid zone. So this is what it looks like. It's right up near the edge of the forest, at the tree line, sorry. And there are these alpine meadows. And within these alpine meadows, here we can play spot the graduate student. So every summer we were taking some graduate students, postdocs, and master students, as well as some interns and volunteers to go for the summer and to be catching these lizards. And this then we would phenotype these individuals. So it's involving a lot of collections of lizards from across the landscape, and of course, maintaining information on where they come from. And yeah, we even use some child labor involved. And so taking this information, there's some variation in the belly coloration of some of the individuals at this contact zone, but in general, in hand, we can't distinguish an oviparous and a viviparous. And so what this means for doing a study on oviparous and viviparous phenotypes is that we do this by phenotyping the females. So we keep them in enclosures for the period of incubation and then record the information about their reproductive success and if they have had live birth or eggs. And so we do this over many, many years and many, many females, and then have found cases of putative hybrids in this instance. And something, just a short digression, one thing that we're doing to try to improve our resolution for the hybrid zone, because we don't know until after the end of each field season whether we had hybrids or not, is that we've developed a diagnostic SNP set that works with something called the Biomeme platform. And we developed this in collaboration with this company who has this on what they call on-the-fly molecular biology. So they have, this is a qPCR machine attached to an iPhone, and the iPhone comes with it. And we can use this to do diagnostic SNP analysis in the field. So we just before we had to stop going because of coronavirus, but in that year we had this up and running. So we were able to do diagnostic analysis of our SNPs, which of course we had determined earlier. So we collect the lizards then in the field. We're doing this kind of on-the-fly DNA extraction just at the lab bench, which was our lab bench, which is really the campground. And and then use this and run it to test so that we can know from the collections very quickly which parity mode we have, and also we can do sexing of juveniles who are too young to be able to be sexed based on phenotype. So using this, the whole setup of this analysis in the field, and we're looking at these alternative reproductive mode phenotypic differences and also looking for hybrids. So we do these collections, as I showed you, in the alpine meadows. Then we bring the lizards in and we keep the females until we see their phenotypes. So they can lay eggs if they are oviparous. And then most of our sampling is as non-destructive as possible, but we did sacrifice some eggs from each clutch to be able to measure the developmental stage. And this is the embryo. It's at a very early developmental stage. And then after a period of external incubation, then they hatch as little tiny lizards. And in the viviparous, what we have is also the collection after mating, keeping the females. And then what they do is they lay these basically egg sacs that have little tiny, you can see one here, little neonate lizards within them. And sometimes there's a little short period of a few hours to a day between them, the parturition, and then all of the individuals emerging from these egg sacs, but there's no calcification. And what we were able to characterize these phenotypic differences in the different parity modes. And what we found, which was really exciting, and I hope would make Werner Meyer happy to know, was that we did find these hybrid phenotypes. And so these are a range of intermediates between the oviparous and the viviparous. And so here would be an example of, in the middle panel, of what seems to be really like an exact intermediate, both genetically and phenotypically. And so what they lay is these very thinly calcified eggs, eggshells, or like patchy calcification. And the embryos within them are also at an intermediate stage of development. And so I should say that this means just not only that these, clutches are hybrid, but it means the female is a hybrid phenotype. And they do, so we characterize these from a number of different aspects. And I just wanted to point out some of the more sort of morphological differences. So we also analyzed the calcium composition and the density. So this row here shows the density of calcium crystals that are found. So in an oviparous, the arrows are pointing, it's quite granular. If you look through an electron microscope, you see this very detailed feature, and it gets more thin and sparse until there is none in the viviparous. And the eggshell thickness we analyzed as well with EM, and could see a dramatic difference between the oviparous and the viviparous. Has, almost nothing to measure. And then we have the intermediate phenotype between. And we also see then looking not only at the phenotypes, but then also looking for genetic evidence is, of course, the key. So assessing genetic evidence of hybrids. We did this using genome-wide RADseq loci that were genome referenced. And what we found is that I'm sure most people are familiar with these sort of plots, but so here each bar is a different individual, and this represents the ancestry as opposed to one cluster or the other. And so you can see we have our very characteristic oviparous genome signature, we have our viviparous genome signature, and then we have intermediate variation between them. Everything from putative F1 hybrids and then backcross in between. And we can link, looking only at the oviparous and viviparous. Is that okay? Suddenly got really big feedback. Did something change? But that's new, right? It was fine before. Okay, I'll speak slowly. Maybe somebody's unmuted or something. So an association between phenotypes. So here, external incubation days. So oviparous have external incubation period, and viviparous are hatched. They don't have this external period. And we can see a correlation. These are each different individual females. There's correlation between genotype and phenotype. So the Q value, as I showed you in the plot before, diverging between the oviparous and the viviparous, and this hybrid span between them. So this was pretty exciting because actually, as far as we know, this is the only hybrid zone that has now been characterized between divergent oviparous and viviparous forms in any amniote. So people had been looking for something like this for quite a while. And here we had our first evidence of this. So we use this then as an opportunity to look at the genetic architecture of this reproductive mode and also gene expression and selections. We'll talk about these next. So how we did this, then we took an approach of admixture mapping, which many people will be familiar with, of course. So we take our parental taxa, so in this case, our oviparous or our viviparous, and now we have our F1 hybrid between them, and then we have many different recombination events that are our admixture events. And now here, instead of doing this in a lab, as would be done in some experimental systems, here our alpine meadows are where this recombination events are happening. So we're sampling individuals and using those as our population to study. And so we did this as a combination of about 80,000 SNPs for association analysis with this admixture mapping. And then we also used 65 individuals, a whole genome sequence to look at candidate genes. And what we found from this was that we were able to map these genotype-phenotype relationships. And so we focused on two we combined traits into two major kind of groupings. So we have gestation time and we have eggshell traits. So this includes a combination. And what we found was that these were distributed in some cases differently and in some cases the same across the genome, but clustered in some particular regions with very high association between these traits and the genetic basis. And we've seen some enrichment for this on a couple of different chromosomes, and more genes were involved in gestation time than we found involved in eggshell traits. And I'm not going to today go into great detail about the different candidate genes because it becomes quite, many different approaches we use for different ways. But we found that some of these genes that were found in our admixture mapping analysis were also found genes that are known to be associated with duration of pregnancy and also calcium trafficking. Then we also looked at, wanted to make an association analysis between the functional genomics of this through gene expression in complement with the admixture mapping analysis. And so to do this, we did RNA-seq analysis of individuals that were taken from the same reproductive period. So early, during, and during pregnancy, and then also immediately post. And we did so with the oviparous and the viviparous females. And we had then an overlap between, we had genes that were in early and mid-pregnancy, and then also some that were after. And these, we found consistent signals of differential expression between the oviparous and the viviparous. And those genes that were differentially expressed after, we removed from the rest of our analysis, and we considered these to be, well, they might certainly have some functions in reproduction. We considered them to be ones that were also a matter of evolutionary divergence between these two lineages. So we don't account for those ones. And what we found is that we had, you know, substantial differences in gene expression between viviparous and oviparous pregnancy. So these are the ones that were upregulated in the oviparous and upregulated in the viviparous. And so there's 2,610 significantly differentially expressed genes. And from these, we found expression, sorry, I should have said that this was from oviduct tissue if I hadn't. We did find expression of genes that were known to be associated with maintaining pregnancy in other species. So of course, this is the trait that has been studied in other squamates and of course extensively in mammals. And so we found genes that were also known to be involved in skink, which is another squamate, and also in mammals and marsupials, for example, and also some that had even come up in seahorses, as well as others that had been involved in other lizards. And importantly, we had an association between our genes that were differentially expressed and the genetic regions that we had found to be associated with between genotype and phenotype. So we had a large number of candidate genes that were inferred from our genome analysis that were also differentially expressed genes. And so this shows this concordance of information and really is another line of evidence for the functional role. So we know that these have diverse phenotypes and that there's differences between these viviparous and oviparous lineage in a number of different traits, and that there's the reproductive trait, but then there's also different, differences associated with these lineages themselves. And so we wanted to explore a bit of information about selection on these genomic signals we had for our different parity modes. But we have also, as I said, about a 3 million year divergence between these two lineages. So it's important to remember that these are coming back in contact. This is not the early stages of divergence in the parity mode, but it's admixture between the divergent lineages. And so when we found looking for genome-wide signals of selection, that these are distributed, all across the genome. And so highly reflective of a combination of neutral divergence and signals of selection. But we did find evidence for overlap between genes that were found to be under selection and genes that were candidates in our genomic analysis of the functional association. And so one way we can look at this to begin to tease apart this relationship, I'm just accelerating a bit, knowing we're a bit late on time because of my tech problems. Sorry. So I'm just going to touch on some of the ways that we can look at this relationship briefly. So one thing would be to look at FST, so genetic divergence, and then looking at regions under extreme divergences as well. And so here we have all of our different genetic variants, and we have a distribution of FST that maxes out at 1. And these are quite divergent lineages. So there's a lot of FST between them. One thing we could do then was to look at our genetic variants that had been associated with gestation time or with eggshell traits and overlay these with the selection analysis, and we see that we do have association then where we could call them QTL or we could call them candidate genes, that these regions were also showing significant signals of selection in many cases, and that we have this distribution of these in some interesting locations across our distribution of differentiation. And so this is something that we're working on more now, more in detail with whole genome data, because as I said, most of these were drawn from RAD-seq data. So now we're doing it more at a nucleotide level. But I just wanted to show that we have a concordance of signal then between selection, functional role, and candidate gene from the association mapping. And then as a last step in this particular study, I wanted to explore the role of these shared genes in pregnancy across vertebrates. And this is actually quite challenging because of the massive phylogenetic difference that we're looking at when we think about repeated origins of viviparity, as I spoke about in the introduction. And then also, noise associated with many different experimental protocols. So the way that to try to get a little bit at the generalizability, so with the aim of going beyond just talking about particular candidate genes or particular, biological pathways that may be enriched in our study, trying to take a bit of a quantitative approach to seeing how this relates to other studies. So using this, multiple different origins of viviparity, focusing then on the viviparous lineages. So we generated a database of genes that were significantly differentially expressed in pregnant compared to non-pregnant tissue in a bunch of other studies. And this database in the end was of 13 species. And then it is a statistical analysis to look for significant sharing between them. And we did this in a couple of different directions, looking at oviparity and viviparity and looking within squamates, but I'm just going to speak about the most broad analysis that we had. So this is just looking at the viviparous common lizards, for example. And we found that there was overlap between these differentially expressed genes in pregnant and non-pregnant viviparous vertebrates. And so these are spanning these across different squamate species that are viviparous, and then also across a selection of mammals. And then as an outgroup, we explored a little bit also with seahorse, which is of course not really viviparous, but it is a case of male sort of analogous to pregnancy, and so has shown some signals of having similar gene expression profiles as actually female pregnancy. And what we found was that there was a significant overlap of these differentially expressed genes across these really massive evolutionary timescales. And of course, these are too small for you to look at it, but in detail. But this is just showing that there are this enrichment of shared genes across many different species. And so for example, just to break this down a little bit, so the top 10 most shared genes, so most intersections, span both mammals and squamates. So this is a sort of shorthand version to say that it's not that there's some genes of pregnancy that were squamate specific, like that the most genes shared are squamate specific, or that they're mammal specific, but that in fact we found evidence across the phylogeny for really deep sharing of these differentially expressed genes, suggesting tapping into really ancestral networks that are shared between amniotes, maybe at some level between vertebrates. So there was much less sharing between the N-amniote representative that we have, but still significant sharing relative to background expectations. So just in summary, so what we had seen then was showing that there's these phenotypic differences between the alternative reproductive modes, characterizing them in detail in order to do our admixture mapping, and also then this existence of hybrids between the oviparous and the viviparous. Now the hybrids are found with quite low frequency, depending on the hybrid that we're looking, whether we're talking about F1 hybrid or putative F1 hybrid or some more extreme back crosses, but it ranges from, it's sort of a few percent between maybe up to 10% of individuals collected. And this is geographically variable. So accumulating the sample size needed for this analysis to quite a few years of hard work led by Hans Recknagel. And we identified and characterized these oviparous and viviparous hybrids and showed how they're intermediate both in genotype and phenotype. And of course, we always have this idea of these hopeful monsters that come from hybridization events. But in the case of these oviparity, viviparity, you know, they're kind of it's in a fitness valley. It's a bit of a doomed hopeful monster. They have we have a I have other data about their success, but we do they are viable hybrids, but only found in this very, very narrow zone. And then we showed this that we had identified the genetic architecture of these major reproductive traits, successfully genetically mapping on this wild population, which was at this single field site. And so we have also looked for other field sites, but so far this is the population that we have. And we found that these parity mode traits were associated with few regions of large effect. Some of them are particular to oviparous traits, and some of them are particular to viviparous tendency traits, and then some of them are overlapping. And these included genes that are involved in pregnancy and calcium trafficking, both in squamates and in mammals, and that we had these different regions for development and incubation compared to the generation of the eggshell itself, which is the ancestral. And then we had functional differences in gene expression between our parity modes, with shared expression relative to diverse and divergent viviparous vertebrates. And our genome-wide signals of selection, and that there is concordance between these signals of selection and genomic candidate genes. We also, a direction I didn't have time to talk about, have signals of selection associated with habitat and elevation. And here we try to tease apart those that are particularly to do with parity mode. And then we found these particular viviparity genes associated across amniotes. And so we've generated this database from this literature review of differentially expressed genes, and so it's available for other people to examine. And so the work that I talked about today was just recently published in the last few weeks in Nature, Ecology, and Evolution, if you wanted to look into any more details about it. And so this is our first evidence for this reproductive mode genes done by a genetic cross. And so the hope for the next steps for this will be that we can use this as a launch pad to evaluate reproductive mode evolution in other species, because now we have a genetic architecture and we have some key candidate genes. And we suggest that this simple, sort of relatively simple genetic architecture might facilitate these repeated transitions that have happened. And in order to test this, we'll need to look across more evolutionary replicates. And with that, I'd like to thank our funding sources for this study and the lab members who are involved, especially Hans Recknagel, for leading this work. And thank you very much for listening.